Hi everyone, as promised, here is the remainder of Unit 7 and 8 on DNA replication, mRNA transcription, and protein synthesis. This combination of units is concluded with a discussion of how gene expression is regulated. Remember that we want to maintain the concentration and the levels of proteins within some optimal range so that they are in a state of homeostasis, right? This dynamic equilibrium. And there has to be some mechanism or mechanisms then to regulate how much protein is being transcribed and translated. What you see here is a, a sequence in E. coli, right, which is a characterized by this thing called the lac operon. So we have these genes that are clustered together. All right. And this region here is ultimately what is going to get transcribed into mRNA by our familiar friend here, RNA polymerase. You're also going to see regions of the DNA here. All right. The template strand specifically of the DNA. Remember that RNA polymerase reads just the template strand, not the coding strand. You see these regulatory regions that are not going to end up being part of the mRNA, or not going to inform any sequence of nucleotides in the mRNA. So you can see here that we have in this template strand of DNA the transcription unit, meaning these are going to be read by the RNA polymerase to make mRNA, or the RNA polymerase to make mRNA. And then you have the regulatory sequences here, which are bound by various regulatory proteins to dictate whether or not this region is going to get transcribed. So what you'll see first of all is this cap binding protein binding to a region called the cap binding site. And the binding of the cap binding protein to the cap binding site helps to activate or promote transcription of the gene. Okay, so it helps the RNA polymerase to bind to this region called a promoter to begin transcribing that template portion of the DNA. On the other hand, right, we have a repressor, and repressors bind downstream of the promoter region in a region called the operator. And the binding of the repressor, as you can see here, obstructs RNA polymerase so that polymerase is no longer able to move along the template strand of DNA to transcribe that portion to produce mRNA. All right. So here we see different scenarios where the presence of the repressor, right, prevents the transcription from occurring, right? But upon removal of that repressor from the operator region, the polymerase is able to bind in conjunction, actually being promoted again by the cap binding protein binding to the cap binding site. So the presence of this protein and the absence of this one will allow for RNA polymerase to bind, and therefore we say that the operon is on and transcription is allowed to proceed. And of course the RNA polymerase is going to move along and catalyze the addition of nucleoside triphosphates, catalyze dehydration synthesis, and as a result string together a strand of mRNA, one nucleotide at a time. Of course, that happens until the RNA polymerase reaches the transcription termination site, at which point the RNA polymerase will detach from the template strand, and we have our pre-mRNA that will go through post-transcriptional modification. Okay. These regulatory proteins are referred to as transcription factors. All right, so these can either initiate, promote, or repress transcription. So now we switch gears and talk about mutations. And mutations, by definition, are changes to the genetic material, specifically the DNA. And errors can arise in the DNA during replication. In fact, the more cycles of DNA replication there are, the more likely that mutations are going to accumulate. There is, however, a mechanism to proofread this so that there are genes that code for DNA repair enzymes. And what they do is they compare new strands of DNA with the original strands to fix the incorrect nucleotides based on the rules of complementarity. So, for example, if we look at this portion of a double helix, again with parallel, antiparallel strands, and our backbone of phosphate and sugar in each of those strands with the protruding nitrogenous bases forming hydrogen bonds between the complementary strands. You can see here that if there were to be an omission, right, a mistake where these thymine residues are omitted, then by token of the presence of adenine nitrogenous bases, adenine bases over here, it's obvious then that those missing nitrogenous bases should be thymine, and so the correction takes place. All right. Now, eventually, these mechanisms start to falter, and mutations will start to accumulate. And there are implications for those mutations because, as we recall, that DNA codes for protein. And if mistakes are accumulating within the code, then the protein synthesis might result in the addition of incorrect amino acids. Because the side chains of amino acids differ in their chemical and physical properties, that can result in changes in the three-dimensional shape of the protein and therefore render the protein inactive, right, non-functional. And it can also lead to something called apoptosis, which is a programmed cell death, or it can signal to our immune system that this cell needs to be destroyed, and so our immune system attacks those. Furthermore, some of those mutations will lead to disease. For example, cancer is often a result of the accumulation of these mutations. Now, mutations can occur at specific nucleotides in the DNA sequence, or they can involve the shifting of entire regions of DNA. Focusing on the ones that occur at specific sites, these are called point mutations, and they can be of one of three types: a substitution, an insertion, or a deletion. And the name of the type of mutation is pretty self-explanatory. A substitution mutation occurs at a specific nucleotide, right? And what happens is that nucleotides are replaced with different ones. For example, an adenine could be replaced with a thymine, or a guanine, or a cytosine, right? A cytosine might be replaced by an adenine, a thymine, or a guanine. So that's a substitution. One nucleotide out, or several nucleotides out, but an equal number of nucleotides back in, but different ones. An insertion mutation is the result of adding nucleotides to the normal DNA sequence. So you're adding nucleotides without taking anything away, and you'll end up with a greater total number of nucleotides in the DNA sequence. The opposite of that, of course, is deletion, where nucleotides are removed from the DNA sequence. And when it comes to insertion and deletion mutations, they can lead to something called the frame shift mutation, because, and this is specifically occurs when the insertion or deletion results in the addition or removal of a certain number of nucleotides that is not a factor of 3. That number 3 is really important because we know that there are 3 nucleotides in the codons of mRNA. So if we add or remove any non-multiple of 3 nucleotides, it causes a shift in the reading frame, what's called the reading frame of the RNA polymerase. And I can show you some examples of this shortly. Recombination mutations involve moving portions of DNA from one location to another. So the overall effect isn't an increase in the total number of nucleotides, it's just a shifting of position of the nucleotides from one part to another. However, that can inactivate the protein because you want the protein to fold a certain way in a certain sequence, and so by moving that sequence elsewhere, right, the folding is the tertiary and the quaternary structures might be negatively affected and compromised. All right. Now, we've just talked about the mutations in terms of how the genetic material itself is affected. But we can also classify mutations according to what the consequences are for the proteins. All right? And there are three types. A nonsense mutation changes the codon to a stop codon, and it causes the ribosome to prematurely detach from the mRNA. If you go back to earlier in the unit when you had that table with all 64 codons of mRNA, you remember that each codon, right, resulted in the addition of a specific amino acid in the protein chain. In that table, right, okay, first of all, let me clarify here. Okay, these are mRNA sequences. And remember that mRNA is transcribed based on the template DNA, right? So the template DNA, let's say it gets mutated to GTT, right? Remember that template DNA is read by RNA polymerase, and a complementary nucleotide is added, CAA. Well, when the ribosome reads this CAA as a codon, that informs the ribosome to recruit a tRNA bringing glutamine. However, if the DNA got mutated instead to ATT, okay, then the resulting mRNA would be complementary UAA. And when you go to the table, right, UAA tells the ribosome to stop transcribing. And because of that early stop, the resulting protein, let's say the protein was supposed to be this long, right, ends up being shorter because the ribosome encountered the stop codon too early due to that mutation. All right. And this can be catastrophic because, of course, a protein that is significantly shorter is going to fold very differently than a protein that is longer. A missense mutation, right, results in a different amino acid being added to the growing protein chain. And that can also change the way in which the protein functions if the changed amino acid is chemically and physically very different than what it was supposed to be. So again, I'm going to specify that this is your mRNA, and this is informed by the DNA template. And so UUU in the mRNA would have been derived from AAA in the template DNA. And if this gets changed by what's called a missense mutation to CAA, then the resulting mRNA from CAA would be GUU. Well, UUU is supposed to code for phenylalanine. If you refer to the table, you can verify this. And if that changes to GUU, then the amino acid that's going to be added is no longer phenylalanine, it is valine. And because of that, because we've changed the type of amino acid that's being added from phenylalanine to valine, this is referred to as a missense mutation. Best case scenario, though, is a silent mutation. And we looked at a specific example where I demonstrated that the redundant numbers of codons coding for a limited number of amino acids allows for mutations effects to be mitigated to some degree. Okay, silenced in other words. Again, let's assume, right, this is the mRNA. This is coded for the template DNA. So this would be A, but if that were to be changed to AAG, the resulting mRNA codon of UUU being changed to UUC is inconsequential because UUU and UUC in the mRNA both code for phenylalanine. So while there was a change in the DNA resulting in a change in the mRNA, there was no change in the amino acid being added. Okay, here is an example. We have an mRNA. Again, the important thing about that is the sequence of 3 nucleotides that form the codon. And CAA, all right, codes for glutamine to be added to the growing protein. Now, if CAA is changed to CAG, right, there's the mutation, right, then that's okay because CAG still codes for glutamine. The resulting protein will not be changed unless there's a mutation in other residues. If CAA is changed to UAA in the mRNA, then that codes for the stop codon. This is a nonsense mutation. Meanwhile, if CAA is changed to AAA, we have a missense mutation because what should have coded for glutamine now codes for lysine. And the consequences for these mutations can be quite catastrophic. Here we see an example of a missense mutation. In sickle cell anemia, all right, there's a particular codon, GAA, in the mRNA that's mutated to GUA. And specifically at this 6th amino acid, right, glutamic acid is changed to valine. And that is enough to denature what is supposed to be this biconcave disc shape of hemoglobin to this sickle shape. And this is really catastrophic because the more surface area we have on the red blood cell, the more oxygen can be carried. And very notably, this lower surface area results in a lower oxygen carrying capacity. These blood cells also break apart really easily. Sorry, they don't. It's not that they break apart easily. They actually are able to obstruct and get stuck in the blood vessels easily, and therefore compromise the structural integrity of the blood vessel. And this can also prevent oxygen delivery to the tissues because if you can imagine this causing that occlusion, that blockage, any tissue that is deprived of oxygen over a prolonged period of time can be severely damaged. All right. Here we have an example to work with. Our DNA double helix will result in the transcription, or is being used in the transcription into mRNA. Okay? And I just want to remind everyone that the DNA doesn't change into mRNA, it's being used to make mRNA. And this is by RNA polymerase. notice that only one strand of mRNA is created, and that's because RNA polymerase only reads the template strand. Notice that CAA AUG is complementary to GTT-TAC, so it's this lower strand that's being read. Notice again that our mRNA is practically identical to the coding strand, with the exception that because this is an mRNA sequence, all the thymines are changed to uracils. And then using that table from earlier in the unit, we can see that AUG, ACC, GGU, UCA, UGC, and UUA code for methionine, threonine, glycine, serine, cysteine, and leucine. All right. If there is, excuse me, if we have a mutated base pair in the normal DNA, so here we go from TA being changed to CG. Right? So notice that change here. This gets changed to this. Right? Then our codon that normally coded for serine now codes for proline. Right? And this is a missense mutation. Right? When a different amino acid is added instead, we have a missense mutation. Now, what if we have an insertion? Okay? When we have an insertion, this causes real damage because here we're going to insert an additional nucleotide. All right? So from here to here, in this spot right there, in this spot right there, okay, A and T have been added, right? So A added to the coding strand and T by complementary base pairing was added to the template strand. Well, what happens now is our reading frame has shifted. Recall, these three nucleotides coded for methionine, threonine, glycine, serine, cysteine, and leucine. Okay, and I want you to keep in mind here, that hasn't changed. Okay? But here, with the addition of this single nucleotide, look at what happens. There's our reading frame. But now, because of the addition here, everything has shifted. Right? AUG and AUG, that's fine. ACC, that's fine. But now instead of GGU, we have GAG. Instead of CCA, we have CUC. Instead of UGC, we now have AUG. Instead of UUA, we now have CUU. So this methionine and this threonine are unaffected because that reading frame has not changed. But because of this insertion here, right, instead of glycine, we now have glutamine. Instead of proline or serine, we have leucine. Instead of cysteine, we have methionine. All right. And by fluke, we still end up with leucine at the end. Right? But look at it, rather than one amino acid being changed in this missense mutation, we now have many amino acids being altered due to that frame shift. So we're literally shifting the frame along. And deleting nucleotides can have a similar effect. Okay, here's a closer look at frame shift mutations. Here's my original mRNA sequence, AAA, UUU, GGG, CCC, coding for the amino acid sequence lysine, phenylalanine, glycine, and proline. This is the reading frame. In other words, the codons that are being read. Okay? And if I have deleted nucleotides, so let's say these two are out, then AAA is red, but instead of UUU, because of this shift, UGG will be red, and that will inform the addition of tryptophan rather than phenylalanine. And because rather than GGG and CCC being red, the next codon will be consisting of GCC, which codes for alanine. Again, if this just keeps continuing on with that frame shift, you have a drastically different protein being synthesized. Okay, we've just described point mutations, right? Mutations that occur at a specific region of the DNA due to addition, deletion, insertion and deletion, or substitution. Now we're looking at recombinant mutations, where we have a segment of the DNA called a transposon that's actually moved to another part of the DNA. Just as our reference point here, we have a normal gene flanked by our regulatory sequences. And again, this is going to determine, how often this gene is expressed. Now, if that transposon ends up within the gene, then we've mutated the gene without altering the regulatory sequences. And this means that the gene is going to be expressed in the cells they're supposed to be expressed in, but due to this addition here, right, the protein is going to be longer than it's supposed to be, because this is going to result in the addition of more amino acids, and the protein is not going to function properly. If the transposon, right, is inserted in the regulatory regions, then that's not going to affect the gene itself, and so the proteins that result will be functional. However, they might not be expressed in the cells they're supposed to be expressed in, and because the regulatory region is being affected and the regulation of the gene is part of how we maintain this in optimal levels, we might not have an appropriate level of this protein. And if the transposon inserts far away from the gene, away from the regulatory regions, and away from the gene itself, then this protein is going to work fine. All right, and it's not going to be affected whatsoever by that mutation. So how do we acquire DNA mutations? Again, I mentioned earlier that with each subsequent round of DNA replication, these mistakes start to accumulate, and the mechanisms that safeguard us against these mistakes start to falter. We have the transposition and movement of segments of DNA from one region to another. We can also inherit mutations, and 10% of diseases are from inherited mutations. So in other words, the mutations occurred in your parents, and that was passed on to you through their haploid cells, or sperm and egg. Certain environmental causes of mutations are mutagens and carcinogens. Viruses can also cause mutations. And in terms of radiation, UV light from the sun can cause mutations. In fact, this is the most common cause for skin cancers. X-ray radiation or gamma radiation using for X-rays can cause several types of cancer. And certain commercial and goods contain chemicals that are harmful to us. For example, certain pesticides and herbicides, right? And hence the move to eat more organic, quote-unquote, organic food. That is food that is grown without these harmful chemicals. Industrial chemicals that go into the making of a lot of the products that we use on a daily basis also contain carcinogens. Pollution, cigarette smoke, additives in food that help preserve them and make them last longer. they may stay quote unquote good and last longer before the expiry date. However, those additives can have cumulative effects, and certain drugs also are mutagenic. In terms of viruses, these are some of the viruses that are associated with specific types of cancer. For example, the HPV virus, which is responsible for genital warts, can cause cervical cancer in women. The HIV virus, AIDS, can cause Kaposi sarcoma. And hepatitis B or C, as the name hepatitis indicates, affects the liver and can cause liver cancer. And finally, Epstein-Barr virus is a form of mononucleosis, which can cause lymphoma, which is a cancer of the lymph. So this is just for your own interest if you ever want to explore this further. But our main concern with the upcoming quiz is on the types of mutations and the consequences they can have for protein assembly. All right. So let's look at this example to conclude the lesson. Here we have as our reference point normal DNA. Again, you can see the anti-parallel strands of the double helix, 5' to 3', 3' to 5', and that the nitrogenous bases are complementary to each other. It says here that the bottom strand is the template strand. So let's just make sure we are aware of this. And we know that the template strand specifically is being read by the RNA polymerase, not the coding strand. So if this is our normal DNA, then the resulting mRNA, right, will be A, T, G, C, A, U, G, C, U, 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 C, A. Okay. So this is now our mRNA. I want you to go to that table and determine the amino acid sequence. All right. Now, if this gets changed, if there's a mutation here, and our adenine is now thymine in the template strand, the resulting mRNA sequence will be AAG, CUU, and UCA. Use that table and make that amino acid sequence. Okay. And I want you to classify if the resulting mutation is silent, missense, or nonsense. Okay. So in this final example, our mRNA will read AUG, CUU, and UCG, and determine the amino acid sequence. All right, so this is the final portion that you're responsible for in preparation for the upcoming quiz next week. All right, take care, everybody.